right, welcome everyone to my oh, the tie is a little tight. All right. Welcome everyone to my master's defense. I'm Shamim Ahmed presenting my master's defense today on the competitive response of cannabis sativa, which is hemp with different agricultural weed species. So it's been 2 years I'm here in SIU in Dr. Gibson's lab. So over the last 2 years, it's been an absolute pleasure working with Dr. Gibson's and working on cannabis. So today, throughout my presentations, I'll give you the overview of what I've done over the last 2 years. And I'll talk about all the study that I've done, both in field and greenhouse setting. So let's get started over here. All right. So throughout my presentations, I'll talk about hemp. So like what hemp is. I know we all know it's a plant species, but I'll give you a little more information. I'll talk about why my study was important in the first place, why I did do that, and then what sort of hemp cultivars that I choose for my study, and which are the weed species that are pretty common and that are significantly affecting the growth of hemp parameters and also their yield as well. And based on my background, I did have some objectives. And also based on those objectives, I did have some hypotheses as well. So to test all the hypotheses, I had 3 different studies. One of them was done in the field setting, which is in ARC pod, and the rest of the 2 were done in the greenhouse setting. And finally, I will talk about what I got from all these 3 studies and what sort of future research we could focus on from there on cannabis. So hemp, so it's basically herbaceous dioecious flowering plants from the Cannabaceae family, which was primarily originated from the Eastern Asia, especially some part of India and China. And there are like 3 different cultivars, especially varieties of the cannabis, which is Cannabis sativa, indica, and Cannabis ruderalis. So first, out of these 3, the first 2 is basically produced for fiber and THC content, and also for CBD content as well. But the first one, the sativa, the level of THC is below 0.3, but in Cannabis indica is basically above 0.3, and it's basically not for the THC level, the Cannabis sativa ruderalis. And then, so the background, why my study was important in the first place. So we all know how much weed species is like significantly affect the growth of crop species. So what about cannabis? We never know. So one of the study that Shikana and Gage they did in 2022, they found that weed species like water hemp, like amaranthus, they affects hemp yield productions. But how about the other weed species? Like, is there any significant effect of other weed species on hemp growth parameter or their yield production in the first place? And also like how weed species, like the study that was done in Shikana in 2021 and Malbada in 2023, they found that weed competition basically creates some kind of stress, competition stress, which basically increases the THC level of hemp, and that turns the hemp into marijuana, which is basically illegal. And we also found that for controlling weed species in the hemp plants, we don't have so far any kind of labeled herbicides. And so even if we use like labeled herbicide, it's very much time consuming, and it takes a lot of money to invest over there to control the weed species in the hemp field. So for my study, I choose 2 different cultivars. So according to the International Hemp Associations, we have over 600 hemp cultivars in the first place. But I choose these 2, which is Jinma and NWG, Northwest Genetics. So the first one on the left side, you could see here how the Jinma variety is much taller and thinner, and it's basically grown for textile fiber. And on the right side, you could see the NWG, which breed for grain, yield, and seed. And it's basically much smaller, shorter than the other variety Jinma. So for the weed species that I've found in my field and from the literature review, these are the common weed species that significantly affect the hemp growth parameters and hemp yield over the growing season. So especially the goosegrass, water hemp, morning glory, and giant foxtail. These particularly 4 weed species that I found in my field experiments and also in my greenhouse experiment, they significantly affected the growth parameters like hemp stem diameter, their height and number of leaves over the growing season, and also the biomass and the relative yield as well. I'll talk about the relative yield and everything in the next couple of slides. And then trigonolene. I'm sure many of you the first time heard this word trigonolene, which is basically an alkaloid derived from the nicotinamide. It's also considered as a drought stress marker, like it can be found in the tuber, in the leaves and seeds of the different plant species. So far, in cannabis, we found it has like some secondary metabolites like cannabinoids, flavonoids, and terpenoids. But how about THC, which is trigonolene? So far, we have not found any kind of study that has detected the THC in cannabis leaf. So I'm the one who first detected, who first just tested whether there is a significant level or detectable level of amount of THC in cannabis leaves. Okay, so where are my objectives here? For 3 different studies, I had 3 different objectives. For the first one, study 1, I quantified the differences in the competitive response of hemp with different weed species to determine whether these weed species and hemp are competing with each other for any kind of resources, like light, water, and nutrient. And based on the first study and objectives, I had 2 different hypotheses. So I hypothesized that there will be no differences in the hemp growth parameters, especially their height, number of leaves, and stem diameter by their weed competitor density. And also, I hypothesized that there will be no difference in hemp nitrogen and carbon assimilations by weed competitor density. And for the study 2, which was done in the greenhouse setting, which is basically also a competition study, but in a different setting. So I quantified that the differences in competitive response of hemp cultivars. For the first study, I chose only 1 cultivars, but for study 2, I chose 2 different cultivars. So that's why I just mentioned cultivars with 3 different weed species to determine if the weed species are competing with hemp for resources like water, nutrients, and any other kind of resources. So as I mentioned earlier, so for the first study, there was no selected weed species. But for study 2, there are 3 different weed species that I will just talk about later. And based on this study 2, I have like another hypothesis for number 2.1. And I hypothesize that there will be no difference in hemp plants growth parameters by hemp and weed ratios. Okay, so the hemp and weed ratio is basically like in mixtures. I'll talk about that later. And for the final study, study 3, which was also done in the greenhouse, I quantified the accumulations of trigonolene as a drought stress marker in the leaves of cannabis. And I hypothesize that there will be no significant differences in the accumulations of trigonolene as a drought stress marker in the leaves of cannabis sativa cultivars. Because for that study, I also used 2 different cultivars of hemp. Okay. So this is my study 1, the field experiment that was conducted in ARC plot, which is Agricultural Research Center in SAU. So if you have a look here, so on the left, it's the actual plot that guys made. We're just measuring the plant's parameters, measurements, and also that's the plot design. So just have a close look over here on the picture, right? The plot design. So this is one of the plots. So I have total 8 plots altogether. So out of the 8 plots, 4 of the plots were treated with herbicide, grass-specific herbicides. Okay. So for each of the plots, so both hemp and weed species, they were co-occurring in the same plot. So what I did was, you could see over here, the heart sign basically indicate different kind of weed species. And the circle, the white circle, is small and big one, they both are hemp plants. So what I did was I randomly chose 5 different hemp plants and tagged them. Okay. And what I did was, so T indicates basically the tag plants. Okay. And I made a 30-centimeter circle around the tag plant. And what I did was, over the growing season, I measured each of the tag plants' height, its stem diameter, number of leaves, and also counted the weed stem density by species. And I also counted the number of hemp other subpopulations that were cooperating with the hemp around the tag plants. Like this is the tag plants and that's other hemp plants. I also counted them as well over the growing season. So as I mentioned, I took data on hemp parameters, their height, stem diameter, and number of leaves. I took data on the weed count, weed stem density by their species. And as I mentioned earlier, so I had total 8 plots. So in 8 plots, for each of the plots, I have 5 tag plants. So in total, for 8 plots, I had 40 tag plants. So I took the data over the growing season of the hemp and the weed species, like their vegetative pre-flowering, flowering, and their reproductive stage as well. So, yep. And once I have the data, so working. Jump to new variables. Sorry, guys. Okay. Sorry. Well, so once I have the data, I ran an ordination analysis, which is a non-metric scaling, multi-dimensional scaling. So this is basically, these two pictures is basically the mirror view of each other, okay? So I ran an ordination analysis for all stages across plots to test the community composition of weed species with or without herbicide. And I found that, so these 8 different color ellipses is basically 8 different plots in 4 stages with herbicide and without herbicides, okay? And I also did a PERMANOVA, which is permutational multivariate analysis, multivariate analysis of variance to see the differences of the community compositions across plots. I also ran an PARM dips to see the dispersions among the weed species community across plots. So what I found over here is like the herbicides by state interaction was significant for PERMANOVA. And here I found that, so if you have a look here, so this is, as I said earlier, these 2 pictures is basically of the mirror view of each other. If you just put it over here, it will be the same thing. So here for height, so height is basically related with the weedy species here, like the Epomea, the morning glory, Cypress esculentus, and Allium vinyl, which is wild garlic. And also I found that the soil moisture is basically increasing in the later stages, like 4Y. So why 4Y basically the late stage 4, and why the applications of herbicide, yes, but it's no over here. I also found that how the weed count is just the opposite of the height. I mean, if the weed count increased, the height decreased, and it was same for the stem diameter as well. So as I mentioned earlier, so basically we can conclude that like the larger hemp height, they have associations with different kind of woody species like Allium, Cypress, and the Cypress Epomea species, basically. And we also found that the height and stem diameter of hemp increased with a fewer number of woody species in the first place. And then I just ran in regression analysis to see whether the herbicide has an effect on weed community. So if you have a look over here, how the presence of herbicide and absence of herbicide affect the weed diversity and the weed count for their growth cycle. So what I found was right here, so weed diversity significantly decreased with the presence of herbicides, okay? But without the presence of herbicide, it's fluctuating over here, but it's reducing over time. And it was same for the total weed count, like how the herbicide affected the weed diversity. So, sorry, weed density. So it significantly reduced the weed density, rapidly decreased. But here, the weed density slowly decreasing over time. And then I try to see how the individual weed species affecting the hemp growth parameters. So these are the all simple graphs. So we have like some full models where we run like mixed effect models. And here we could see here how the hemp height were significantly affected to morning glory, water hemp, and goosegrass. So in the next slides, the full models where you just run the mixed effect models. So you could see over here how the effect of interactions significantly affecting the hemp growth parameter, especially the height. So if you have a look here, the interactions of number of hemp around the tag plants, the subpopulations, herbicide and stage significantly affecting the hemp height over here. So here you could see here, just imagine conditions like you apply the herbicide. So for the grassy species, so you think, okay, so the herbicide is supposed to reduce the number of grassy species, which means it's supposed to increase the hemp growth parameters. But here, you could see here in stage 3 and 4, the height is decreasing. And it was same for here as well. So what is the reason behind? So we assume that, so even though the number of grassy species reduce over time, but there could be possibly other broadleaf woody species like water hemp, morning glory, which ultimately affected the hemp height and reduced the hemp height over times over here. And looking at my data straight, I found the same thing here. So the summary of my field experiment is I did the similar kind of analysis on the other hemp parameters like stem diameter and number of leaves, and I got similar kind of results over time here. So the first hypothesis was rejected based on my results over here. We found a huge differences of the growth parameters of hemp, their stem diameter, height, and number of leaves over time by their with competitive density. But it supported our second hypothesis. We didn't find any kind of differences in the nitrogen and carbon assimilations of hemp by their with competitive density. So we summarize that the hemp performance is pretty much related to the environmental variables like soil moistures, species diversity, total weed count. And we also found that herbicide usage is basically was effective for controlling the grassy species, but it possibly increased the relative abundance of the broadleaf woody species like morning glory, water hemp, and some other woody species that we found in the field. All right. So this is my second study. As I mentioned earlier, my hypothesis that the ratio of wheat and hemp. So here, it was done in the greenhouse using a DU wheat replacement series, which means like there'll be different ratios, but the equal total population density there will be. So for this particular study, I choose 2 different cultivars of hemp, that is Jinma and NWG, I already talked about it, and 3 other plant species, water hemp, giant foxtail, and soybean. So out of these 3 woody species over here, these 2 are basically woody species, but soybean, it has some agronomic value in the first place. That's why I chose that one. So here you could see here, I germinated both the hemp and all the other woody species in the flats, and then I transplanted them in the pots, just like over here. This is the pot size. And I had total 120 pots, 2 hemp cultivars, 3 species, 5 different ratios, and 4 replicants. So the ratios basically indicates, so in each of the ratio, there will be mixtures of the plant species. There could be like 100% hemp, 0% wheat, and then 75% hemp, 25% wheat, 50% of each, and then 25% hemp, 75% wheat species, and then 0% hemp and 100% wheat species. So as I said, so there are like 120 pots for each of the replications for total 4 applications, and then total 120 pots. So, and then I collected data over the growing seasons, like in the vegetative stages. They are in the vegetative stages, pre-flowering stages, flowering, and then reproductive stages. So I took measurements on hemp height, their stem diameter, and number of leaves. And I took measurements on the wheat, their height, number of leaves, but no stem diameter, because they're very thin to just measure the stem diameter. So, and then I also collected all the plants and took the biomass of each of the pots biomass, like based on their mixtures in the pots. So I ran an ANOVA, a 3U ANOVA, and also ran an 2K HSD, and saw that the total biomass, wheat biomass, and the hemp biomass, they were significantly affected, especially the ratio that you used and also the wheat species. So if you have a look over here, the hemp biomass increased as the proportions of hemp is in the mixtures increased over time. And for this, wheat biomass was highest for giant foxtail especially. So we can summarize that the wheat biomass is depending on especially the individual wheat species. And for over here, the total biomass in the pots, like in each of the pots, there was like some kind of mixtures, either wheat species or hemp or both of them. So we found that the ratio significantly affecting the total biomass. And here, the total biomass increased only when the wheat in the mixtures increased. And we also ran the same kind of analysis on the hemp yield, relative yield, wheat relative yield, and also total relative yield total. So what is relative yield in the first place? So it's basically the comparison of performance of strains in the mixtures relative to its output at the same total population density. And the relative yield total is basically the sum of the relative yields of the two strains over here. So I did the similar kind of analysis for all of these HRY, which means hemp relative yields, wool relative yields, and the relative yield totals. And I found that the ratio, either ratio or weed species, or both of them, were affecting the relative yields of hemp and weed species, and also the relative yield totals. Now, so as I mentioned earlier, so I measured the growth parameters of hemp over time. So I ran an ANOVA and 2K HSD again for those as well. So and I found that how the interactions of ratio and stage are affecting the height over here. And I found that over stages, the hemp height increased. And for these, the ratio and the weed species interactions are affecting the height. And we found that especially hemp plants are much shorter when they're growing with the soybean than giant foxtail, regardless of the ratios. And then we also found that the interactions of hemp cultivar and stages, it was affecting the hemp growth parameters, but we found that hemp increased through stages over here. And interestingly, we found that sometimes the hemp cultivars, like there is a variations of hemp cultivars height. So we found that except stage 1 and 4, the Jinma cultivar of hemp was always taller than the NWG cultivars, okay? And I ran the similar kind of analysis for hemp stem diameter, number of leaves, wheat number of leaves, and its height. So, and I found a similar kind of results. All right, so I already mentioned the relative yield total. So here you can see 3 different graphs, each of them for giant foxtail, water hemp, and soybean. So what I did over here, I got the relative yield of both hemp and the individual weed species and the relative yield total. So what I found over here, so our expectation was the relative yield total is supposed to be 1. That's our expectations. But if it goes over 1, it's over yielding, which indicates that they're growing better in the mixtures. But if it goes below 1, they're not growing better in the mixtures, rather they're growing better in the monoculture. And the hemp and the wheat relative yield is supposed to be exactly their percentage in the mixtures. Well, now I'm going to just give you descriptions over here. So if you have a look over here, you can see the hemp and wheat together over yielding in mixture at 25, 75, and 50-50 ratio. So what does that mean is, so the red line over it signifies with the standard error bar that in a ratio of 25, 75 over here. So it's supposed to be, as I mentioned earlier, it's supposed to be 1. But if it's here, we can see here how it is like above 1, which means they're over yielding. And it was the same for 50-50 ratio over here as well here, right? You can see here it's over 1. And then if you come at wheat at 75-25, where is basically 25% of the wheat species right over here. Like the blue line, the green line here signifies that it's basically over 0.25. So here it's over 0.25, which means it's over yielding in the mixtures. And it was same for the hemp alone at 50-50 here. You could see over here how it's like over 0.5 because like the ratio is 50-50, which means the hemp relative yield is supposed to be 0.5. If it goes over 0.5, it's over yielding, and it was happening here as well. And for the hemp alone, it's under yielding at the mixture of 75-25 because at the 75-25, the relative yield is supposed to be 0.75. But here, you can see here, the relative yield is basically below 0.75 right over here, which means it's under yielding in the mixtures. So I got similar kind of results for the water hemp and the soybean as well. And sometimes it's showing like both hemp and wheat species or individual hemp and wheat species, they're either over yielding at some mixtures, and sometimes they're under yielding for both the water hemp and the soybean as well. So Marginal studies summarize that we found the result and that rejected our hypothesis because we found a huge differences in hemp growth parameters by their wheat competitive density and also for the wheat parameters as well. And we've also found that the ratio especially in the wheat species significantly influences the relative yield total of hemp and wheat species and their relative yield totals. And we also found that the hemp height was quite shorter when they're growing with soybean than the giant foxtail, regardless of their ratios. The final study. So that was also done in the greenhouse, but in a different bench. So as I mentioned earlier, trigonolin is a derived stress marker in the leaves of Cannabis sativa. So we so far haven't found any kind of a study that has done so far. They found the trigonolin. So we're the first one to detect the presence of trigonolin in the leaves of cannabis. So what I did was I chose the same cultivars, Chinma and NWG, and I just germinated them in the flasks and then transplanted them in the pots, both Chinma and NWG. There was no weed species, only these two cultivars of hemp. And so 2 hemp cultivars, 2 treatments, and 10 replications, to total 40 pots, 20 pots for each cultivar. So I let them grow, these 2 cultivars, once they were transplanted, I let them grow for 45 days. So after 45 days, I started the treatments. I separated them. Half of them were under control. I mean, half of each cultivar were under control treatment and drought stress treatment. And then after 5 or 7 days, I collected data. So how did I collect data? So I extracted leaf, okay? So what I did was I measured both the relative water contents of the leaves and also the level of trigonolin. So what I did was they were grown in the control environment, which is in the greenhouse, and then I extracted the leaf for each of the pots. As I mentioned earlier, I had total 40 pots, right? So I had 40 vials for RWC and 40 vials for TRG, which is trigonolin. So what I did was I extracted the leaf and just weigh the fresh sample and dry them for 55 degrees Celsius for 48 hours. And then I just found, I'm putting them in the vials. I just calculate the relative water contents. And then I also had 40 vials for separate 40 leaves, okay? Where I just measure the trigonolin, just putting them in the vials with methanols, distilled water, and chloroform in a ratio of 5:2, and then I just quantified and purified them using spectrophotometry. I again purified them using an ion exchange chromatography. So once I had the data, I ran an ANOVA, and where I found that both the level of trigonolins and RWC showed a significant differences between the control and drought stress treatment. So if you have a look over here, the stress plants had higher level of TRG, which is very much important. So, and this is basically the log value, because we had some the skewness in the data. So we had to just transform into log, and then we found that the treated plants with the stress plants, they had higher level of TRG. And here we found that the level of RWC were lower in the stress plants, which is very much logical. And we plotted a graph, trigonolin versus relative water content, where we found that in TRG over here, in drought stress treatment, the level of TRG, the volume of TRG is increased 3-folds compared to the control treatments over here. And it's the whole graph signifies the level of TRG and RWC for each of the cultivar over here with their standard error. So as I mentioned earlier, both the TRG and RWC show significant differences between the control and drought stress treatments. So we summarize that the drought stress increased the accumulations of the TRG in hemp plants, and it supported our hypothesis. Hemp cultivars basically did not affect the TRG accumulations, rather the treatments did. So what was our findings based on all this study? So for the first study in the field experiment, we found that the later stages of the hemp growth parameters were significantly affected, possibly by the broadleaf woody species, because in the later stages, we found that they were significantly affected by the broadleaf woody species. And then the competition between the hemp and the wheat, it doesn't create any kind of deficiency in the total nitrogen and carbon assimilations that would increase the level of terpenoids and cannabinoids in the hemp plants. And for future research, so for both of our study, the greenhouse and the field setting, for the field setting, we use direct seeded hemp, but for the greenhouse, we use the transplanted hemp, right? So we could just focus on measuring or comparing the vulnerability between these two kinds of plants, like between the direct seeded hemp plants and also the transplanted hemp plants to their weed interference. And for the second study, we found that both the interest-specific and interest-specific competition reduced the relative field of both hemp and weed species. And for the findings, we could focus on finding any other cultivars because we have a lot of cultivars of hemp. So we could just focus on finding any other hemp cultivars that is more competitive than those particular hemp cultivars to controlling the weed species. And for the third one, study 3, we found that the drought stress it does influence the accumulations of trigonolin, but the cultivars of hemp didn't have any effect on the trigonolin accumulations. So we could just focus on finding, is there any other hemp cultivars that could have or might have differences in TRG accumulations? So these are my references. So now the acknowledgement. So it's been over 2 years, almost 2 years, I would say. I landed in the US in fall 2022, August 9, stayed the night, first night in Chicago in a hotel, the country where I have never been. It was like, it was very much adventurous for me, staying in a hotel right in Chicago the first night and just come Carbondale the following afternoon. And then coming from a country 8,000 miles away from the US, it was never been easy for me in the first place. Everything was new because that was my very first plane journey outside country and coming to the US and getting acclimatized over here. It was never been easy, but the people over here in the US, all the people that I met over the last 2 years, like they're super fantastic. I'm truly grateful all of them. So first of all, I'd like to thank my advisor, Dr. Gibson's. Dr. Gibson's, I'm really lucky and privileged to be a part of your lab over the last 2 years. I just can't really be thankful to you enough for all the guidance and support and all the kindness you showed over the last 2 years. And I am really, truly humble and grateful to you for all the things that you've done for me over the last 2 years. And then my committee members, Dr. Gage, I don't know how to express my gratitude to you because for all of my study, the field study, the greenhouse study, the input and the insights that you put, the way you just pushed me to just go beyond my limitations and everything else, it was amazing, to be honest. I have grown as a scientist, like, well, maybe I did something, but now I know how to do research in the first place. So thanks for all the things that you've done, and thanks to your lab members, Christiana, Eric, those who helped me out in my field experiment, also in the greenhouse, and also Alison as well. And then Dr. Leme, really thankful for all the inputs in my proposals and all the things that you've done over the last 2 years to excel me in my research and everything else. I'm really, really grateful to you, and also for the recommendations that you just provided me for my PhD admissions. Thanks. And Dr. Andrew, so I did most of my work outside lab, which was also in the field, but through my trigonal experiment where Dr. Wood helped me out throughout the experiments, it has taught me some kind of molecular level work that I had no idea of in the first place. So Dr. Wood helped me out throughout my trigonal experiment, provided insight and everything else. I'm really grateful to you, Dr. Wood. Thank you. And Gibson lab members, I know, Brian, you were here in the Zoom, maybe on Zoom. As I said, I'm really grateful to you. So these two guys in the pictures, on the right is Brian, on the left, wearing the glasses is Ken. So the Brian was always there for me, giving me a ride to my lab, sometimes setting up my experiment in the greenhouse, helping me out to collect data, and what's not. Like, he was always there for me for everything, even in like writing my thesis, analyzing my data. Even when I just asked him about any kind of like help or something like that, he was always there for me, even though he has had a lot of stuff to do in the first place. I'm really grateful to Brian. I'll remember you for the rest of my life for the things that you have done for me over the last 2 years. I just can't really be thankful you to enough. And Kane, what else should I say about you? This guy and his wife, Morgan, the Carrington family, they are my local guide over here. And their family member as well. So it feels like they're my home, I am from home. So I'm really grateful to all the support, both mentally and some other way you provided over the last 2 years. I just really grateful for all the things that you have done. I still remember, so I was like mentally disturbed for whatever reasons. And these are the two guys just knocked my door. Hey, we are here for you, having some food, my favorite food from the restaurant over here. So these little gestures, it's very much to me. And Ken was always there for me to just get rid of snacks because I just hate snacks and scared of snacks. And he's the guy who just loves snacks a lot. Even I remember I was just he and I were collecting data in Barker Bluff and Denison Hollow in Harrisburg, and we just encountered deadly copperhead snakes over here. We just escaped it by, I don't know how, maybe we're lucky. And the next following week, we are just collecting data in the hemp field, and I was just identifying a weedy species, which is copperlip. And I was just said, hey, I got a copperlip. And he said, it's copperhead or something like that. So yeah, so yes, they're always for me, like to help me out to collect data and everything else. I'm really grateful to the Morgan and Kane family and everything else. And I would like to thanks to all the people at SAU, ARC, TIC, and HRC, especially Hunter, who was always there at TIC to maintain the temperature and the light we had to just maintain all the time throughout. And I'd like to thank all my colleagues in the School of Biological Sciences. Sheila is here, Rachel is here, Becky's over there, and all the nice people who just always welcome me with like a beautiful smile and the welcoming gestures. I'm really grateful for all of the peoples. And finally, my family and friends, many of my friends and family members on Zoom maybe. So you guys are hearing, listening to me. I'm really grateful to all of you. You guys are the people who have made the sacrifice over the days and years. That's the reason why I'm standing here today and almost there to finishing up my master's today. I'm really grateful to you. And before coming to the US, just 1 year before I lost my dad in 2021. And here, the support that I got here and everything else. So I know like my father just couldn't get it to see to just shine in my life and in my career. So I'm really thankful to my father because the sacrifice he has done over my life and for the days and over years, my mom and my siblings and everyone else, my friends. So these are the people who just made me who just made the sacrifice over the days and years to just make me smile. So I'm really grateful to all the peoples, all the family members out there. So yeah, thank you.